examining the historical evolution of mental health services in India, offering valuable insights into the nexus of science, medicine, and societal responses to mental illness. Without further delay, I would like to invite Dr. Jain to take the floor and share his valuable insights.
I just request everyone to sit down so we can begin the talk. Second, please sit down. If you sit down, everyone will. And actually requested all mobile phones on silent, please. And also remember, we are streaming this, so whatever you say will be heard. We are streaming it live on Facebook. Don't make noise.
So thank you everyone for inviting me here, and like I said, I've been here a couple of times, and it's always good to find like-minded people doing the right things for the kind of people we see, and it's not always. So I retired last year after whatever 40 odd years at Humans, and where I met many of the friends here. They go, and I go back a long way to the 80s almost, and so we all learn psychiatry in different ways through our initial interactions. But what I'll be talking about today is the kind of work that we've done in the last few years. Now, why do we do this? Because as one can understand, the advantage of living in modern societies is that everybody grows up, even people with disabilities grow up. And what happens to people with autism as they grow up is a very different thing. So there are very few studies, but in the clinical world, we have often observed that patients with autism end up having a significantly higher rate of psychiatric disease as they become older. Not necessarily of the full syndrome, but some of the behaviors as adults would be classified as psychotic symptoms. Of course, the basic symptoms of autism, of poor social intelligence, poor rapport, are also better now. Should I speak closer? Okay, I have to stick very close to the mic. So many of these symptoms are. Many of the symptoms that are seen in autism are often seen in adults. Difficulty in social interaction, communication are the basic symptoms of both schizophrenia and bipolar disorder. And therefore, and we also know that there is a big overlap with neurodevelopmental disorder and with epilepsy. So there is a whole spectrum of overlaps. And our preoccupation in the last 20 odd years is that diagnostic syndromes in psychiatry have outlived their utility. There is no point in trying to specifically diagnose schizophrenia, mood disorder, OCD, because very often the drugs you use are several. There is no strict correlation between the diagnosis and the disease. So might as well reorganize our thinking about psychiatry itself. So don't think of it in syndromes as separate diseases, but think of it as a continuum which occurs in different people at different times. And therefore we felt that studying the genetics of schizophrenia and bipolar disorder with specific reference to autism would perhaps be of interest to everyone. Why is this? Because the neuropsychiatric syndromes are by far the most complex and the most unknown. We know that unless you do all this clinical evaluation and the details of matching the clinical phenotype to the genotype, you're not going to get very far. And as you know now, the OMIM has more than 5,000 categories, about half of them are neuropsychiatric diseases. And the Nomad often gives you all this data. But as you can see those little squiggles that are made, the behavioral changes of Parkinson's disease, Huntington, dementia, FPD, ALS, neurological syndromes, movement disorders, ASD, seizures, and neuromuscular disease, these are the neurodevelopmental overlaps. And they form the biggest chunk of so-called unknown genetic causes. So there is a lot to be understood as to why these things occur. And within that, there is a large amount of multi-system involvement and malformations. And of course, involved errors of metabolism, which are comparatively rare, but also can present autism-like symptoms. This occurs for the very simple reason that diseases of the brain at various levels. One is that the head and neck, the CNS and the head and neck have very complex genetic biology, and a large proportion of genes are actually expressed specifically in the head and neck and the brain. Looking at it from purely from not information network paradigm, all the symptoms that we think of in psychiatry, like motor delay, like thinking, like neurodevelopment, like speech, and various other symptom clusters that we think of as syndromic, are actually interconnected. So biologically, these are interconnected networks which co-regulate motor movement and speech, or gestural communication and emotion. So there is no point in separating this out in our heads in the real world. Of course, the genes associated with IDD and with ASD and with epilepsy tend to occur in genes which are much larger. And as any biologist will tell you, the larger the gene, or Parkinson's law itself, the more complex the machine, more breakdowns will happen. So merely the fact that the genes in the brain are larger than many other genes, they tend to accumulate more mutations and therefore more variability. Now whether how that impacts on disease is a different issue altogether. So I'll start with 2 or 3 clinical vignettes. This was a young child. I didn't see the child myself because we had to send our virgins to see them. Because there were 4 siblings, who had all developed normally till age 8, then developed ataxia, epilepsy, and by the time they entered adult services, they were homebound. They could not walk. And 4 of them, they had initial child psychiatry files where they had been diagnosed initially with autism or disintegrative psychosis, because they grew normally till 8 years of age and then suddenly started losing their milestones, and with movement disorder. So then it turns out that when we sequenced them, they had this very rare mutation in the ACNJ10 gene. Then we asked the family, they had another branch of the family living about 50 miles away, which was also consanguineous, where they had the same mutation. The particular mutation that we discovered, which is marked in pink, has not hitherto been reported in the world. It's a novel variant. The syndrome itself includes a large group of, including renal dysfunction, but our patient did not have renal dysfunction. So the position of the variant in the genome gives rise to different symptom clusters. And when we looked at and impaired the sodium pump in every cell, and we even got some cell culture work done in our lab, and the people who carried the mutation had a clear-cut deficit in the sodium channel function. So you could go down, drill down this all the way. But it tells you that rare variants, hitherto unknown, may be present in the community and contribute to a very rare syndrome of what otherwise seems to be a complex phenotype of disintegrative psychosis, movement disorder, epilepsy, which is progressive and leads to very severe disability. This is a more typical thing. This is a very 19-year-old person who has been defined to have ADHD, query, and autism very early, with oppositional behavior disorder and conduct symptoms, and anacastic personality traits from the mother, schizophrenia in the maternal grandmother. And he had very severe skin picking, obsession of contamination, pathological doubts, checking, contrast behavior, needed symmetry. He was a very orthodox vegetarian and would not go out at all because the risk of stepping on chicken bones. He wouldn't allow you to wear leather. He wouldn't allow his sister to carry a leather handbag. So it was extremely severe. And he was quite, on examination, he was quite obese, and multiple capital spots, and calculomania in the chest, and inguinal tracking. The clinical diagnosis was neurofibromatosis, but the genetic diagnosis, and clinically had OCD with ADHD and opposition defiant disorder. And we detected these variants. Now, it turns out that the position of the variant in the neurofibromatosis gene can predict whether a child has autism, ADHD, or has brain tumors. So this particular child's variant was in the front half of the gene. It's most commonly, it's the most commonly mutated gene in humans. And in this particular case, the mutations in the first half of the gene are less likely to have severe autism symptoms than in the second half of the gene, and the tumors and the autism seem to go cross over each other. So this particular child had this variation in a particular part of the gene. Neurofibromatosis is a modern system to understand autism because it interferes with lots of synaptic pathways and second messenger pathways. And you also happen to carry a variant in the trio gene, which is otherwise known as a variant of unknown function. But the trio gene also interferes with GTP exchange factors. So both on two different parts of this pathway, he had a mutation in the neurofibromin gene and in the trio pathway, of which regulates GTP production. So in that sense, this progression, this particular variant was quite informative. He is not getting better at all. He's been on clozapine, but a little bit of adding of statins improved a transient benefit in the symptom because statins and the cholesterol pathways can also regulate this pathway. So you can now get a genomically informed pharmacological decision. This is a much more recent case. This is a pair of twins who had been extremely shy and schizoid. They hardly spoke to anybody else, spent most of their time talking to each other. They were academically not too bad. They finished their school, but they always had very poor social skills. For example, after going to somebody's birthday parties, they would, as they call, what's it called, when you intrude into somebody's selfie, they would always stand behind and do that. They wouldn't know that they're not supposed to be there. They would blow out the birthday candles instead of letting the other person do. So they had a little social awkwardness. They didn't accumulate many friends. And as they grew older, at 12 or 13 years, they started having these body image disturbances, skin picking, preoccupying with looking at the mirror, insisting on surgery for their eyebrows or the nose, etc. And then they started having typical schizophrenia symptoms, talking for 2 hours and things like that. So all this happened over 5 or 10 years, and they came to us, and they had very brisk deep tendon reflexes, and they had a restricted range of emotion, reduced psychomotor activity, empathetic affect, poor insight, and they would spend most of the time talking to each other. MRI had bilateral intensities. They also had a mild cardiac defect. So it turns out they had a variant in the POGS gene, which is one of the rare but better defined genes that are linked to autism. And it modifies chromatin structure, interferes with mitosis itself. And this, according to some researchers, and I'll show you a slide, this is a distinct subtype of PSD, which does not necessarily have to involve ID. There are some normal carriers who have the mutation, but large majority of them have autism type features. About 50 cases are known in the world as of now, that's all. But the interesting part is that the gene has been biologically well characterized. So these are the previously identified reported variants. These are the more recent ones, below are the more recent ones. And you can see the various syndromes. You have heart disease, you have ASD, you have developmental delay linked to different positions in the gene. So depending on where the variants are, you get a different kind of syndrome. You get a different kind of syndrome. And the protein itself has a number of zinc fingers and DNA binding domains, and it regulates mitosis, and especially in the neurons, it's very critical for its function. You want to extend the mic?
No, Sanjeev, you can actually hold that down and you may be more comfortable with that.
That's fine. I was thinking of knocking and creating a noise. And the expression is high during early embryonic development and decreases gradually after birth, remains constant. And in a Drosophila model, it impairs learning in the fly. So now you can create a Drosophila model for autism, which is also very nice, because then you can start testing out various methods and how to correct that defect, either through stimulation or by other mechanisms. So these learning defects, which you can model by producing specific defects in a specific gene and understand the biology, therefore offer a unique approach. And here again, the pathogenic mutations affect the second half of the protein. So there's a whole lot of overlap. Now this takes us back to the basics of psychiatric genetics, that there are a number of, in many instances, probably the risk of psychiatric disease is linked to both ends of the spectrum. Certain individuals or families have high penetrance, high impact, rare variants, and a large number are a mixture of common variants. But these people who have a kind of multiple affected Mendelian kind of inheritance, low frequency variants, intermediate penetrance, can be very usefully understood by whole exome sequencing or by whole genome sequencing. And these kinds of cases, over our experience over the last, let's say, 5 or 10 years, if there's a very strong family history and overlaps with both ends of the spectrum, with early onset ASD and or developmental disorders, atypical presentation, unusual pattern of treatment resistance, or overlaps with neurodegeneration, like early dementias. I won't be talking about the dementia cases now, but both with neurodevelopment and with neurodegeneration, there may be an overlap of psychiatric syndromes.
Let us
So,
ask,
sure.
how can Drosophila model of autism relate?
The patterns of learning and attention can be mimicked in the Drosophila. The reflex responsive or adaptation. It's not the whole model, it's just a symptom of habituation which can be modeled in a Drosophila. So, and even if you can have a partial overlap, that's good enough. So we started with a few families, 8 families, with lots of, with multiple people affected, sometimes 5 or 6 people affected in the same family. Work done mainly by Suhas and Aditi, whose Partha had guided her PhD work years ago. So these were particular families. And I won't go you with the details right now, because I'll come back to that. But there were 42 variants, of which about 1/5 were also were found in genes that are previously linked to a known OMIM syndrome, known OMIM neurodevelopmental syndrome. So 1/5 of the patients had variation in genes that we know cause developmental delay or autism. This is a very large family that we were lucky to collect over many years, which almost 100 people, history available for more than 100 people, 40 or 50 people affected with mood disorder, DNA available for about 25 of them. And again, the genes involved were to do with ankyrin repeat domain on chromosome 1 and cyclase gene involved with actin cytoskeleton on chromosome 6. So these kinds of, and none of these is the biology well understood at the moment. So in the next sequence of exome analysis, we looked at 75 families and looked at 78 variants. We detected 78 variants. Now these variants were described as with the usual definitions. They have to be deleterious by 2 or 3 or 5 paradigm, shift, polyphen, etc., etc. They have to be very rare, below 0.001 in the population. And they obviously should not be shared as far as possible between affected people and unaffected members of the same family. So you get lots of variants, and you can, on the list is alphabetically whatever dense, but that's not really relevant. But you can see that overlaps in neurodevelopmental syndrome, cerebral hypoplasia, cerebral atrophy, neurodevelopmental disorder, impaired intellectual development, and autism come up highly in the list. None of the patients had so-called defined syndromes of autism. These are all adult patients who were coming to routine psychiatry OPDs. There was no prior history of autism specifically in these individuals, but we did pick up these kinds of genes. And now the proportion implicated in Mendelian disorders has increased to almost 1/4. When you looked at the specific syndromes that they were involved in, and you can see that they are involved both in neurodegeneration and neurodevelopmental pathways. So there is neurodevelopmental and cerebral atrophy or spongy degeneration, and microcephaly, epileptic encephalopathy, hypoplasia, and autism comes up again and again. So these are the specific syndromes that we discovered variants in, which were also, these are the OMIM syndromes in which they were engaged in. We also looked at whether synaptic biology itself is implicated, and when we looked at the gene-wise burden, then we found found that there are particular genes which tend to carry a lot of variants, which is the ones up there, right up to the POTTE gene, and the ABCD1, which is just reaches significance. Most of them are involved in neurodevelopmental pathways. So we also want to look at synaptic genes, because by now there was a very interesting paper from the South African native population where they defined 1200 odd genes which are implicated in synaptic biology and looked at rare variants in those genes compared to the normal population. So we had a so-called ready-made list of so-called genes presumed to be involved in synaptic biology, and they were also carriers of slightly higher rates of mutations. Now this obviously creates an issue, because how do you understand the, for example, the constant rates of schizophrenia and the ever-increasing rates of autism? Our genomes aren't changing that fast. So why is the clinical syndrome changing fast? And that's been a complete mystery. Now it turns out that schizophrenia and dementia may actually be in a state of balanced selection, that there may be some advantages to the schizophrenia genes which protect against infectious disease similarly as for dementia, and therefore there may be some trade-off in this. And now for this particular thing we looked at schizophrenia-related genes, and we found that two sets of genes, mainly the immune-related genes, showed greatest evidence of selection in the families with schizophrenia, even more than the native population. So there is some evidence that there may be some trade-off between infectious disease and brain disease that seems to be playing out. But this obviously needs confirmation in many other settings. So these are the kinds of papers that we've been publishing for the clinicians. So the kinds of exomates that you get, what you understand of the biology, what you understand of the clinical presentation, and various kinds of complex syndromes that can occur with one or two variants. But I won't be describing these. These are mainly to do with neurodegeneration. So what is it with autism disorder that we want to do? This is the work we have we've been lucky enough to get young faculty under the DST Inspire program, Ashita, who's finished a PhD in St. John's on autism. Now we know that all these facts, it's birth to a lifelong disease. Globally, 1 in 100 children have ASD, and prevalence is growing for unknown reasons. And the difficulties, as I mentioned, are to do with social communication, etc., etc. Now, what are the CNV studies that we have? Because these were done years ago, because we have lots of capacity to detect CNVs fairly rapidly. But all these cumulatively account for just 1% of the disease. And some CNVs recur at the same locus amongst unrelated patients. So perhaps there are hotspots or instabilities that are at play. So this is happening over and over again. But sometimes it's absolutely de novo and there are not shared healths. Many of them focus on the neural synaptic complex, and they often occur with other neuropsychiatric and neurodevelopmental CNVs, suggesting that there are overlapping pathways. Now, from whole exome studies, which have been slightly more energetic, mainly because of the Simon Foundation and all the other Safari databases, there's lots of data which is accumulated all over the world, with again focus on synaptic-related pathways, glutamate signaling protein, cadherins, vesicle proteins, and ion transport channels. But again and again, these are very seldom large enough or don't have enough diverse populations. So we don't really understand this too well completely. But we know that certain aspects like synaptic functions, chromatic modifiers, embryonically expressed genes, and translation control, if you go back, POGZ gene fulfills all these criteria. So that's why even a few cases of even one gene may be very interesting for understanding how autism occurs. Now we know in general that like with psychosis, there will be large number of common variants. There may be some merit in doing GWAS studies. There may be a finite number of de novo variants, and there may be environmental causes. Now, for various reasons, the GWAS studies on autism have been quite rare in India, because nobody has got sample sizes which are large enough to actually do this. So we are lagging seriously behind. The European studies and American studies have sample sets in tens of thousands. In India, I don't think it doesn't even reach 1,000. So GWAS studies are therefore quite implausible in India at this point in time, unless we really make very specific efforts. But all these other rare inherited SNVs and de novo CNVs have been reported in case reports off and on. And you'll recognize lots of names from this center here, because lots of single gene studies were done from this institute over the years. So many of these studies have been done here over the last 2 decades. So all the MAO and the various glutamate receptor, glycoprotein and various other markers have all been investigated in many populations, including in this place and at NIMHANS. So Dr. Ashika focused on looking at trios and trying to identify de novo variations. He was able to collect 25 trios, very well studied, and then we started on that. And so this is the inclusion criteria that she had: speech delay, language delay without severe ID, and age of diagnosis not more than the early life of 2-3 years of age, and less than 12 years. And they should have excluded all the known causes of Fragile X, Asperger syndrome, Rett syndrome, etc., etc. So patients who were in that sense not yet diagnosed, and of course severe MR was excluded. And in that, in the whole exome data, this is again an exceptionally dense slide, but just tells you that many of the genes that were implicated in other Western studies like Shank, the SCN2A, the ChRNA7, did end up having variants in our sample also. Many of them were novel variants. So in the same gene, she was able to detect enough number of novel variants, and the passages and the usual things of ion transport and laminins and immunoglobulin genes were involved. So in general, when she used the same list of 1,000 odd synaptic genes, and she wanted to look at whether those pathways were more destructive in autism. And then again, you found that there were more than 3 de novo variations till date in the world literature, and these are the functional categories. So there seems to be some overlap with genes implicated in synaptic biology in conferring risk for autism. So this was quite interesting for us, and this poster is being presented at the WCPG in Montreal now. What was the behaviors linked to these genes beyond synaptic biology? And you can see the entire symptom list of almost classical autism symptoms: impaired learning, abnormal latent inhibition, behavioral withdrawal response, decreased aggression, abnormal discrimination learning. So all these psychological traits, in addition to the molecular traits, were also linked to these genes. So therefore it becomes interesting, and the P levels are there. So one can understand that there may be there may be a genetic component to all these symptoms clusters by themselves. Now, then we went ahead and looked at these networks from the world databases into our databases, because we now have whole exome data, the ADPS, WES, from about 450 people with patients with schizophrenia and bipolar disorder and all that. So the first group of rows is autism spectrum disorders plus IDD plus ADPS. So what are the variants that you get? And you get variants in these 4 genes. As you can see, compensator syndrome, ID with autism and macrocephaly, epileptic encephalopathy, and IDD. So variants in these genes are found across these data sets, in common across these data sets. The next one is the schema, which is the schizophrenia whole exome sequencing over 100,000 people, plus a neurodevelopmental disorder, plus ADBS. And again, you get some kind of overlaps between ataxia, Visceral syndrome, and neurodevelopmental and IDD syndromes. And the last one is with bipolar exome sequencing and neurodevelopmental and ADBS. So, and there also you get a few. The overlaps in the first 2 categories of autism and schizophrenia are much higher than those with bipolar disease, suggesting that there may be some distinction between these. So what does it mean? That so you can read the columns: neurodevelopmental disorder versus non-syndromal neurodevelopmental, Safari, which is the autism database, and the non-Safari data set, and how significant are these in autism spectrum. Obviously, the Safari syndromes are overlapping largely with this, with each of them, and when you come to with autism, it obviously remains high. When you add schema, stays high for the safari syndromes and neurodevelopmental syndromes. For bipolar disorder, it drops sharply. So that you can see that the top 500 genes show significant overlap between both neurodevelopmental disorder and autism. And the ones which are common with safari genes and with schema typically lay in the top 100 autism genes and the schema genes. So there is a significant overlap there. Whereas the bipolar disorder and the neurodevelopmental did not had some overlap, but they did not belong to the top 100. So there is some gradient of risk there. The synaptic genes themselves form a network, which is quite understandable. So both glutamate receptors, cholinergic receptors, and GABA receptors all get interlinked in the groups of genes that are causing the syndrome, and therefore there may be a need, including fMRI, the Fragile X fMRI1 gene right in the center, and neurofibromin NF1 and PTEN. So you can see many of the genes that we are detecting are all becoming parts of one larger network. We also want to look at how these genes are expressed in the brain, and so we started doing database searches. So it turns out that bipolar genes are the ones that are implicated in bipolar sensitivity are predominantly expressed in astrocytes, whereas the ones with autism and the neurodevelopmental disorders are mainly expressed in neurons, followed by oligodendrocytes and etc. Whereas schema and the overlaps between neurodevelopmental genes are mainly expressed in neurons, astrocytes, and oligodendrocytes. And they showed some kind of overclustering. So between autism and neurodevelopmental disorder, there is a co-expression across 25 brain regions and 9 developmental time points. So that we know that there are gradients at which you can interfere with development. There are time periods in which neuronal development and plasticity is occurring. The gene expression is varying at different time points. So if you can identify these time points from your genetic search, then you know when to target therapy and what kinds of therapy you can target by using this information more intensively. So this kind of, so these are mainly in the orbitofrontal cortex, prefrontal cortex in the neonatal stage, and then in adolescent age in the ventrolateral prefrontal cortex and the primary motor cortex. So you can understand that these are periods in which motor skills are developing, or the framework for these is being laid down in the brain, and understanding that therefore becomes very important. When you look at the schizophrenia genes with the neurodevelopmental disorders, it was almost identical to that with autism. Okay, the average gene expression across spatiotemporal points were lower than that for autism, but were in the same set of genes. So the schizophrenia genes are slightly less emphatic than the autism genes, but they are affecting the similar processes in the brain, as far as biology is concerned. And for bipolar disorder, this wasn't true at all. So again, you're getting some distinction. Average gene expression across this time were much lower than for both autism and for schizophrenia. So what does this tell you? That there are processes in the brain that are unique. So biological pathways that the first one is for autism, and the next one for schizophrenia. Last column is for bipolar disorder. The first one is obviously nervous system development, depolarization and calcium opening, synaptic signaling, PPIs, wind signaling, and circadian rhythms. And of course, this overlaps with Rett syndrome, axon guidance, and LICAM interactions and cardiac conduction. You remember both the patients that I talked about, both have cardiac disease. So what are the biological processes that are involved? The common ones of imitative and vocal learning, which we know is impaired in autism, brain development, and assembly and organization, and responses. The molecular functions and the cellular components are also listed. So now you can build a hierarchy right from the psychology to the molecular biology as to why the reasons for the overlap occur. We have no ways to explain these overlaps at the moment, but at least you're getting a kind of, that old cranking cinema hall, you get some view of the cinema. You're not getting the whole cinema at the time, but if you change, you're getting one view of the whole changing spectrum. So that's the state we are at. I mean, we have very few windows to look at, but whatever windows we do have, you should start using them. Because then eventually it will all merge into full-scale Amitabh Bachchan production. So what are the major conclusions that we have? The autism spectrum genes showed significant overlaps with both neurodevelopmental disorder and the safari genes, which was quite high, which is the autism group, followed by schema gene, which is the schizophrenia group. The bipolar disease genes overlap significantly with neurodevelopment, but not with the autism data set. And the brain co-express network genes were found mutated in our in-house ADPS data set, that's our families with multiple members with psychosis, and carried deleterious non-synonymous variants which are shared between autism and schizophrenia. So again, the kind of occasional rare report variants that we were talking about may actually be occurring at a much larger level, and we need to develop methods to analyze that, to understand that in more detail. The autism and the schizophrenia genes and neurodevelopment genes are mainly expressed in neurons and therefore affect neuronal viability and function. Bipolar disease genes were mainly involved in astrocytes and neuronal maintenance rather than others. The autism and the schema and the neurodevelopment genes shared several biological functions overlapping. One was the chromatin histone modification, which means they are involved in the very basis of tissue specialization and cell specialization, which is the basic job of chromatin and histone modification. And they seem to implicate both neurodevelopmental and synaptic activities and at a behavioral level in learning. So these groups of genes impair learning in a particular way, which is to do with imitation. Now this is very interesting because imitative play or imitation is a very highly, there's a recent review I think in Cell which shows how imitative play depends entirely on the nature of the mother. At 6 months, to initiate imitative play correlates with the child's IQ at 18 months. So if the mother mimics and makes baby sound to the child learns how to imitate, and that strengthening of the loop therefore improves neural connectivity and actually improves performance at 18 months. So imitative play, responding, making baby talk, doing gestures, doing meaningless talk actually is good for the brain. And so that kind of imitative learning is very, very critical. Now you could stretch the point further and say this actually corresponds to the oral phase as defined by Freud. So was Freud right after all? So that's a separate metaphorical conjecture, but the neurobiology of it is also quite interesting. So at this point now we are reaching a point where you can actually, at least in your imagination, merge Freud with neurodevelopment, which is I think is a pretty nice space to be in psychiatry at this point. So it's very interesting. Whereas the bipolar genes were mainly involved in neuronal stem cell migration and glial development. I'm not showing you the slides of that, but we did a very interesting study where we grew neuronal cells from those with bipolar disorder. And like all good Indians, we drew a border between them and said they should not migrate. Because that's what you like to do. But turns out that all cells will behave like humans, they will migrate. But the cells from normal people, so-called normal people, migrate in a straight line. Whereas those with bipolar disorder have a very, very stochastic movement. They wiggle and ultimately they reach there, but the time taken to make that movement is much larger and exposes them to all kinds of other neurons because they're not traveling in a straight line. And we could do that in a petri dish. And the person and the cells were derived from a person who had particular kinds of defects in the actin gene and in some gene which is involved in the extracellular matrix. So now you can see that cell migration therefore becomes a crucial part of the risk of bipolar disorder. And similarly with glial development, because glia are being maintained all the time to keep neuronals healthy and they are peculiarly sensitive to excitatory overload. So glial development and maintenance becomes very critical, and again we could show in a cell model that patients who responded well to lithium, their cells were multiplying at a different rate, they were renewing themselves at a different rate, and they were differently prone to cell death. So lithium could rescue cell death in those who were responders, could not rescue cell death in those who were non-responders. So now you are able to look at a pharmacological model in the dish to understand why drugs act and why some people become non-responders. You could also show this for sodium valproate. There were also a large amount of protein-protein interactions which were between autism and schizophrenia genes which were not seen in bipolar disease. Now what is it, what do we translate this into clinical bedsides? But typically, genetic medicine has confined itself to Mendelian disorders, the OMM categories, where the laws of Mendel are maintained to some extent. But laws of Mendel are laws of Mendel, and the clinic is the clinic. So Mendel can recognize a gene and say, or we can identify a gene 100 years after Mendel and say this gene causes this disease. But any clinician will tell you that no two patients of Huntington disease are exactly alike. No two patients with Duchenne muscular dystrophy are exactly alike. So there's a huge amount of variation which is dependent on the genetic background and the environmental situation in which the gene is being expressed. So therefore you have a mixed syndromes, that there may be a gene of major effect and there will be a number of other genes, ataxia genes. Ataxia, for example, SCA3 can present with Parkinsonism in Africans, can present with ataxia in white people, and can present with dementia in another population. The gene is exactly the same, but depending on the ethnicity, the syndrome is quite different. There's a whole lot of mixed things. But now, this recent review suggests that there should be something called a transitional disease in which there may be genes of large effect, which may or may not actually produce disease unless there are a large number of ancillary smaller genes or some environmental stressors. So lots of us may end up being carriers of their variants. Just like with the POGZ gene, there are 4 or 5 people known who are apparently completely normal in their studies, whatever they have looked at, or the siblings of those patients who are completely normal who still carry the variant. So you can keep debating whether this is US, is it variable penetrance or something else, or is there something unique in that particular individual's collection of a rare variant plus other variants in their genome, which will actually produce a syndrome. And deciphering that will take obviously a lot more time, but that's something that we need to understand. Why is it important? Because even though you may not be able to have a drug for the disease, you may be able to interrupt some of the pathways that you detect. So pathway-dependent diagnosis may be much better than trying to make these, shopping bag kind of diagnosis, that this person has this disease and this will work. What you need to understand is in this person, these networks are disrupted at the psychological level, at the biological level, at the network level. And treat the networks and the biology rather than this mythical thing that we call a disease, which is good for the textbooks and for teaching and passing MD exams, but in the real world is not actually very useful. So using the genomic information to fine-tune your interventions, whether they're psychological or logical or rehabilitative or drugs, may actually be a better and a more effective way to treat patients rather than just a very simple diagnosis. After all, we know how this information has changed breast cancer. It has really transformed cancer treatment to a very large extent. Should we at least try to attempt that in neuropsychiatry? Learning from those principles and saying, yes, this person has these things, can we try this, can we try this, can we do this? So now that we have the tools, we should be able to do this much better. And the need for this is very critical because, as you all know, the rates of autism are increasing. Autism is increasing by 3-4 times in the last, let's say, 25 years. My worry as a real psychiatrist is 10 years from now, will the rates of schizophrenia increase? Even if 20% of this turns into psychosis, and that's a conservative estimate, we are then going to have a large number of people who have got pre-existing autism, who now have schizophrenia, whom we are going to perhaps mistakenly treat with all kinds of antipsychotics, make them more prone to extrapyramidal side effects, metabolic syndrome, because autism anyway have eating disorders and gut issues, and we are going to have a medical problem, in addition to all the other behavioral problems. So it's very, very important that we understand this transition, that we follow up these children, we understand what happens to them in their lifetime, what happens to them as they grow old. Unless you understand the entire spectrum of how the genes affect the phenotype across a lifetime, we won't be able to use all this technology, all this insights better. So facilities like this, which actually serve the community for decades, are a unique asset. Because you have these patients, you have these families, you have contacts with the family, and you can actually see the effects of your intervention, informed by all this information. So so it's a very new phase of psychiatry that we're talking about now. We may or may not be able to treat the mutation, but we should be able to treat the background, and we should be able to treat the symptoms better. So that's the way I would look at psychiatry going in the next few decades. Much of this work is the work of a large colleague, a set of colleagues. This program is a bit old, I think 2 years old. So many of them have moved on, but most of them are still there at NIMHANS. And of course lots of colleagues all over India and Bangalore, mainly Bangalore, but also Delhi and ISI and various other people that we've been privileged to work with so many years. Thank you so much for your attention.
It was a very thought-provoking talk. Thank you. So I have a couple of questions. I'll ask one by one. So could you find any sex-specific genetic architecture, like in any of those disorders, spectrum of disorders?
No.
You is a sex-specific. And you have analyzed all somatic mutations. Is there any scope to study germline mutations as well? That would be difficult. Okay.
But that would be very difficult for us to. That would be very difficult for us to accommodate. But there are colleagues at ISI or at NIVMG who's looking at sex-dependent transmissions using GWAS.
I'm also interested in that field. Yeah.
So that those methods are being developed. They're using some of our data,
Okay.
and you may have the answer in about a year from now.
Okay. Thank you. Thank you.
So given the neurodevelopmental disorder genes and the schema and the autism, so do these individuals then have a propensity towards dementia?
We have no idea because nobody has done those long-term studies of autism. Nowhere in the world are there extreme long-term studies available for autism.
It's very interesting what you said about NDD and immune response, because anecdotally I have been observing it in several patients the whole time, even though they were exposed. They have less infectious diseases, and I just correlated after you spoke. But a lot of what you spoke is resonating with progression of dementia.
Right.
And autism as a baseline, which is not defined autism, is more dormant, but expresses later as a neurodevelopmental disorder.
Precisely. That's exactly the point that we are trying to understand.
So when you say we are treating them with psychotic drugs, antipsychotic drugs, etc., this is giving me a little fear. What do we do then? How do we treat?
Oh, we develop new drugs. We develop methods to understand therapy in a more targeted way. That these should be given at this much time rather than for years on end. We try to, basically we need to understand how much and how long to treat, as much as just the drug itself. Right now, the principles of drug treatment in psychiatry are very, very limited. If you had behavioral symptoms for more than 6 months, the American Psychiatry Guidelines suggest treatment for the rest of your life. Now, is that actually true? Is it true for a 10-year-old as much as it is true for a 30-year-old? We don't have the guidelines. So it's a very anomalous situation. None of the drugs we use in psychiatry are technically to be used in people below 16. Okay? But in the real world, you find lots of children being given antipsychotics and antidepressants. And in India, particularly ECTs. India is one of the few countries which refuses to follow the safeguards against ECT. Most countries you have to have 3 psychiatrists agreeing to give an ECT to a child. In India, only 1 psychiatrist can order ECT for a child.
In this long history that I have seen in Manovikas, I have not been an active participant here till recently. I remember earlier cases had more schizophrenia and epilepsy than what we see now.
Okay.
Now it probably comes under the ASD, but also Downs, as you know, is going down for various reasons. But I worry that where did our diagnosis change? Where did the antipsychotics change? And are we doing the right therapies?
I don't really have an honest answer to that. I really don't, because we did a study with the child psychiatry department trying to look at genes those who had autism with epilepsy, those without epilepsy, because there is a big overlap. The problem that happened was that not only did we not get enough numbers of patients, it was also very difficult to get adequate amounts of DNA from patients. Because that time we needed blood samples.
Even now, very difficult to extract.
But now the technology is so much better that you can just make do with saliva samples. You don't need blood samples all the time.
That's much better.
That's much better.
Saliva than it's-
Yeah. So technology is improved in a way that makes these kinds of studies actually much easier to do.
Yes, Yali?
Thank you, sir. That was really insightful. I'm very curious. You have been talking about this, the overlap between the psychiatric, in terms of symptoms between the neurodevelopmental and the neurodegenerative. But if I know it correctly, there is excessive firing in case of the neurons. Sometimes in the case of neurodevelopmental diseases. On the other hand, the neurons are getting lost in the neurodegenerative diseases. So I'm curious about the psychiatric symptoms involved, like are those depression or social withdrawal kind of things?
Right. So this idea was developed in the 19th century. Hewlins Jackson, of the Jacksonian March, and the Todd's palsy, that era, pointed out that after excessive firing, for example, the epilepsy, patients often had a negative state of paralysis of the arm. So after the excessive firing, there was a state of almost lasting 2 or 3 days in which the arm would be paralyzed. So excessive firing was very often followed by cell death or cell dysfunction. And excessive glutamatergic signaling may actually cause enough cumulative oxidative damage to alter the synaptic biology over a long term. Which is why, at least on the bedside, we try to explain to people that withdrawing drugs for epilepsy, for example, is very bad, because in a single episode of cluster seizures, you may burn up a lot of neurons. Okay, so controlling epilepsy is much safer than trying to do without. Same thing can be extrapolated almost to psychosis. The time spent in florid psychosis often correlates negatively with the extent of recovery. So if you spend 6 to 8 months having active psychotic symptoms, the outcomes are much worse than if it's 6 to 8 weeks. Okay, so excessive firing has a trough effect later. Now, what the biology of the firing and the trough is, we have no idea. Is there a sensitivity or a proclivity to excessive firing, which in itself has the seeds of degeneration? We have no idea. For example, there are very large epidemiological studies which show that patients who ultimately develop dementia also have slight microcephaly, or head circumference itself is lower, right at the beginning. So the very long-term effects of brain volume, brain size, brain biology, or risk of disease at different age periods. Medicine is an ongoing research, so unless you build up these databases, you'll never have the answers.
Right, that's really interesting. I have just another question.
Sure.
So you also mentioned about the resistance to diseases in schizophrenic patients. So we just overcame a pandemic, like a disease, and do you think that this trade-off will have some impact in the long run? I mean, because you mentioned trade-offs, right? We do.
Yeah, but I can give you, it's not covered in this, but I can give you 3 or 4 random examples. The random examples are that after the episode of the 1918 epidemic, 100 years ago, there was a syndrome of what was then called von economoencephalitis, which resulted in Parkinson's disease. For those of you of my age, you may have seen a movie called Awakenings with Robert De Niro. So he was talking about that Parkinsonism which started after the influenza epidemic of 1918, and people were left with chronic schizophrenia, with catatonia and chronic schizophrenia. Not only that, the children who were in utero during the epidemic produced a spike in the prevalence of schizophrenia 20 years later. So the effects of the viral infection on unborn children produced a slight increase in the incidence in the diagnosis of schizophrenia. Now, middle of this 20th century is very troublesome, diagnosis keep changing, so that's an issue. More recently in America, when the first wave of deaths occurred, you must have seen those pictures of new graves being dug in Rikers Island and unmarked coffins being put in. Those are invariably the homeless people, of which 60% are mentally ill. Okay? So a lot of mentally ill people died because in the first wave of the epidemic. But when they looked at data in more detail, they found that if they had been on antipsychotics, the rates of death were actually much lower. So patients with mental illness who were on antipsychotics had a lower death rate, and this prompted us to do a small study with Shantanu Singh Gupta at IGIB, where we tested this and we found that chlorpromazine, because it's a lipid-soluble protein-binding drug, actually interferes with coronavirus uptake into the vesicles where it replicates. And it significantly reduced viral load in cultured cells and in hamsters. By the time the experiments were done, the COVID epidemic was over, so we could do a clinical study. We can't wait for another epidemic. It's like for our doctors. And hope that we'll get a better research paper, but you never know with doctors.
Thanks. I think we can.
Yeah, when you were talking about this now, in the post-COVID era, if you look at in the last 1 year, there are a number of symptoms. They said that it's a brain fog which is coming up.
Yeah.
Now, this brain fog, and when you were talking about earlier about the LF1,
Yes.
there are a lot of similarities in there.
Yes.
So, is that actually related with someone?
I have no idea, because I mean, I'm far away from the nuts and bolts of immune biology and my thinking in that sense. So I really don't know. But we do know that patients who have so-called viral infections, whether it's viral fever like influenza or more severe like encephalitis, the brain recovery can be very, very long. Encephalitis can give rise to very prolonged severe disability, and even those who recover without apparent neurological deficits, at least they don't have focal deficits and no scarring on the brain, still take years to recover their full concentration ability. So there is obviously an element of inflammatory responses which affect the astrocytes and the glia, which takes a much longer time to revert. And the impact, I mean, it makes common sense. The neurons will live for 6 months, and the glia will be changed much more often, and the inflammatory response will now be acting on a substrate which lives very long. So common sensically one can anticipate that those inflammatory processes which will die away quickly with the mucosal membrane will persist for much longer in a long-lasting cellular tissue like neurons. But whether it's scientifically proven or how would we scientifically prove it, I have
no idea. You mentioned about this RAS and that's triogens actually.
Yes.
So these are the two things also actually could correlate with the difference because that's what we could see in the literature. It's coming up in a very, very big way actually right now when people are looking at it. And my second question is regarding the epigenetic part.
Right.
Because in the first two cases I mentioned about the histone modifications and others. So how far study has been done in this respect?
So my colleague Dr. Meera has been doing this work for quite some time now, and she's right now in the process of analyzing some of this data. At NIMHANS we have something called a brain bank. So we have brain tissue available both from unborn fetuses till 80 years of age. So she's trying to develop an epigenetic clock. What are the methylation changes which occur as a normal part of growing up in the brain tissue, at least in 5 different areas of the brain. So it obviously seems to have some patterns. There's a whole lot of epigenetic modification in the first few years of life, and then another way which occurs during adolescence. And then there's a gradual change. But the data is still being analyzed. There's somebody from Calcutta who's doing a whole PhD on it, and he's come back to write his PhD from somewhere in the UK. So when he finishes his PhD thesis, I will let you know. But it's a very interesting issue because not only is there epigenetics, for example, with the Huntington gene, the blood may have 40 repeats, but the brain may have 400 repeats. So there's a whole lot of somatic mosaicism. And then again, a colleague at IGIB had shown that the brain actually accumulates somatic mutations in a tissue-specific manner, which will then complicate everything that we know. Because if there are genes which are involved in DNA repair and chromatin, and those then make brain DNA more prone to have errors, and then once those occur, then the response to drugs is therefore even further compromised. So that is going to be another level of complexity. But it's something that we'll have to live with and try to understand. We've also done a study on epigenetics of alcoholism, where we try to look at how fast the epigenetic markers on alcohol metabolism enzymes reverse after abstinence, and how are they correlating with relapse and also with propensity to develop cirrhosis of the liver. Because alcohol affects two major tissues, the brain in terms of addictive behavior and the cirrhosis. The two don't always go hand in hand. The cirrhosis of the liver and the brain changes don't go hand in hand. So probably there are separate epigenetic mechanisms in the brain versus the liver which control this. So these are the nuances that we are trying to look at.
Yeah, I have two observations and questions. One is you mentioned about the zinc finger proteins. So it's a little dangerous because these are oftentimes standardly repeated. It's not just one gene. It's a gene family.
Yes.
So one needs
So
to
it is the zinc finger protein binding region in that gene.
Right.
It's not a zinc finger protein itself.
It's not a zinc finger protein per se. The other is that you mentioned about the location of the mutations. What one probably needs to do, and many other people are actually doing it, is to find the location of that particular mutation in the three-dimensional structure of the protein. Oftentimes it's on the surface, when it can interact more with the receptors and so on. But if it's internal or internalized in the three-dimensional structure, probably its impact would be lower.
So we tried doing that. A couple of years ago, we had a student who did just this in terms of GPCRs, where all these drugs bind dopamine and serotonin receptor genes. But thankfully, last year, I think Google decided that all this technology can be used very freely, and so we did this protein modeling. Now that's where the issue comes up. So, for example, that's another paper that we published last year, or 2 years ago, with Parkinson's disease. So, PNH is a very rare young onset dementia, Parkinsonism, dementia complex, Parkinson's type 14, which often starts with depression and schizophrenia. So, we had a couple of patients who had developed schizophrenia at 15, 16 and Parkinsonism by 25, turned out to have PA26. But there the mutation was exactly at a point which it didn't appear. But then, and this was homozygous, the number of people who had heterozygous variants. So then we went ahead and did this protein modeling. Turns out that the site of the mutations affects the binding site of dopamine and also for antagonists of dopamine, namely antipsychotic drugs. So people who have these heterozygous variants, and we looked back at their case notes, were also more prone to have extrapyramidal side effects. But these heterozygous variants are not rare. These are common variants in the population. So unless they are given antipsychotics, they are inconsequential. But if they are given antipsychotics, they will develop severe EPS. So now there is a different aspect to this, to documenting the variants in genes and their impact on protein as also a marker for side effects and pharmacogenomics, not just for disease. So it becomes, and this we can't do unless we do the kinds of things that Pastor talked about. That we have to watch the effect of the mutation on the protein and see how the function of the protein is getting affected. But since we don't know the function of many genes, that creates a problem.
My last observation is that you mentioned that there are about 1,200 synaptic genes, right?
No, in that list.
In that list. Okay. There are more probably.
There are more probably in that list. In that list, which was used by Marie Claire King and Sosa, they had 1,200. This was published in Science. You said if you just
take those 1,200 and map it on a pathway, not all of the genes will be. You can actually figure out some sub-pathways in that broad, large pathway. I'm just wondering whether these different kinds of spectrum disorders and associated other syndromes. If you map the genes that have been mutated or found to be mutated and associated, if you map them on a pathway, do they map on different sub-pathways or do they map just randomly?
No, they don't. So a young colleague is doing this work with SKB's help because gets access to much larger computing space. So they do fall into bins. They're not random. And just to test that out, we've also looked at a number of random, usual things of randomization and trying to see whether they would fit into any other pathway, and they don't. So even the genes that we detected in the WS, when we did all these pathways, if you did a test for how many of all the variants that we detect, how likely are they to be lying in what is the OMIM head and neck syndrome list? And there, you can subclassify that. So they were more likely to be in the head and neck group rather than in a liver GI group. So there is some very broad level partitioning happening. We don't know the fine level detail, but we are beginning to get a much bigger picture. Now, to do the granularity, we need much more work.
But also bigger sample sizes, perhaps.
Yeah, bigger but also unique sample sizes. Just numbers won't matter. To understand this in detail, see, for example, what we call minor physical anomalies. The dysmorphic features that you get on faces of patients with ID. Typically you do a scale, or the clinician says, eyes are far apart, hyperterrorism, blah, blah, blah. But with Digi Yatra, you can do it on a computer. get a Digi Yatra, get a map.
Actually, Digi Yatra is a great way for
It's a great way. To actually classify facial disorder, Digi Yatra is the perfect tool.
Everyone's photograph on Digi Yatra looks.
No, but because they do it badly. You do it well here with all the coordinates, you may actually get some correlate some facial development and correlation with facial genes that regulate head and neck development. No harm trying it. It's free.
So I think we've ignited all our synapses, and now there are multiple questions, but maybe we can continue the questions over lunch. Just want to give a big thank you
Thank
to
you very
Dr.
much
Jain for
having me
being
here.
here, for giving us your time, for sharing all of this, and our kids have a little gift for you. Because we also want to thank you for encouraging us to continue our work for the next 50 years.
I'd just like to add, you are the first event we are having in our 50th year. So we complete 50 years of the institution which you met my mother. She started it in February 1974. I remember I was in college. And today, 50th year is going to end. Thank you.
Thank you.
He has made the painting himself.
Very nice. Very nice. Now. Thank you.
So this is our first event for all of you here. Please remember you have to come to 49 more events. So please please join us for lunch downstairs. Our cafe is not fully ready, but we are hoping this is the run-up to the getting ready cafe. So please join us for lunch.